I don't know if Pratham is here.
I'm
Oh,
here.
he's here. Okay. So I think Pratham will start this off.
So, my name is Pratham Samarth. So, Samarth and I worked on the brain's navigation system, which uses place cells, grid cells, speed cells, and head direction cells to tell the brain where you are in an environment. Uh, do you want to say the project goal?
Yes, the goal for this project was to understand how place cells use the outputs from grid cells and head direction cells to enable navigation, and all of those terms will be explained shortly.
So,
Bounders,
yeah.
Boundary cells are active at the barriers of an animal's environment and are also known as border cells. They emerge early in development before other spatial cells to allow for a vague understanding of an animal's environment that slowly increases in clarity as it grows.
So speed cells are not, they have non-binary activation. So they don't just fire and not fire, they fire at a certain level. So the level of activation basically represents the animal's velocity. So speed cells are used in combination with head direction cells, and this allows an animal to determine its relative position if it knows its start location, speed, and head direction.
The head direction cells are the ones that perform the task that their name suggests, and they activate differently based on the direction of the animal's head. They're found in the thalamus and the hippocampus, and they can be pictured as a ring of neurons where the activity level or the firing rate of a given neuron is based on how it's positioned relative to the direction that the animal's looking at. This bump in activity, as seen on the graph that's on the slide, is the peak firing rate of the neurons in a certain direction, where the x-axis is degrees in this case. And this bump will be important later on.
So this is the ring attractor model equation for head direction cells. So um, the specifics of the equation aren't super important, but the key idea is that each neuron receives input from all other neurons on the ring, which is weighted by their connection strength, which is basically the distance between uh, like the distance between two neurons on the ring. So these weighted inputs are summed together. So the signal times the connection strength. And then if a neuron is near other neurons that are firing strongly, its total input will be higher. So we can see this on the graph of our model, which shows a clear bump of activity centered around the neurons that represent the animal's current head direction. So there's around between -1 and 0 radians. So we can see this on the graph. So also another thing is the orange line represents uh, our simulation with 100 millisecond intervals, and the blue line represents it with 50 millisecond intervals. So we can see the orange line is more accurate because it's wait
Yep.
yeah.
So onto grid cells. Grid cells are output. The grid cells output spatially organized in hexagonal grids, which you can kind of see from the picture. They activate at many locations in an environment to different degrees, so they're not binary, similar to speed cells, and they're located in the entorhinal cortex of the brain. So specifically with grid cells, there's modules of grid cells, and a grid cell module is a group of grid cells with the same spacing and orientation, but different spatial phases or offsets. This collective activity of the module forms a 2D space, which can be mapped onto a torus, as we'll explain in a minute, because it's periodic in both X and Y. So from the two diagrams, you can see on the left, the grid cells are near each other physically, and so their outputs are part of the same module because you can kind of see that they're evenly spaced compared to the green and the pink one, where they're physically distant. So then their outputs are also not similar, and so they're part of different modules.
So place cells are neurons in the hippocampus that uh, their activation is based on an animal's. They fire at a specific location in an environment. However, They don't have consistent firing patterns between environments. So when an animal goes from one environment to the next, the firing location can change completely, which is a phenomenon known as remapping. So the place cell can fire at a different location and at a different activation level.
So as mentioned earlier in the meeting, all of our projects were based on a Nobel Prize. Ours specifically was based on the winners of the Nobel Prize for Physiology or Medicine in 2014, which was won by John O'Keefe, Maybrit Moser, and Edward I. Moser. And it was for a topic that is very deeply related with what we're talking about.
So place cells fire at one specific location in the environment. So this slide is to help you understand how they all connect to each other. So place cells fire at one specific location. Grid cells are like periodic and fire at multiple locations in a repeating hexagonal grid pattern in the environment. They're connected to each other, and when you go from one location to the next, like the connected grid cell fires. So grid cells make up modules that are, and then the modules make up place cells. So together they create a spatial framework that tells your brain where you are in an environment.
So at this point, we have an understanding that head direction cells are a ring of neurons which output a bump of activity based on the direction of the animal's head. And in order to model grid cells specifically, we must add additional input to take that graph from 1D to 2D, or not graph, but output model. So in this case, the inputs will focus on our head direction cells for orientation and speed cells for speed, which allow the animal to find its position. But there are other inputs into this. The shape of the torus on screen, or the donut shape, will be explained in a minute. As explained previously, grid cells are part of modules, and so the output of not three, but multiple of these modules are what feed into a place cell for it to activate.
So on the donut diagram, you can see that there's two axes. So the red one is the first axis and the blue one is the second axis. So they're both periodic because they go in a circle. So that's why the grid pattern is repeating. And it's two-dimensional because there's two axes. So grid cells combine input from head direction cells and speed cells to update a neural representation of the animal's position. So this internal representation is periodic in two dimensions because it's a circular axis. And the resulting activity can be mapped onto a torus shape where both spatial dimensions wrap around. So you can think of if you have a sheet of paper and you connect both the top and bottom edges and the left and right edges, then it will form a torus shape. Yeah.
So, as mentioned, place cells take input from multiple grid cell modules, multiple toruses, so they have a non-periodic mathematical representation. This means that they only activate at a specific position, unlike grid cells, which are dynamic. And so this means that these are kind of landmarks to where you are in a given environment.
So this diagram shows how grid cells map to place cells and also where they are in the brain. So grid cells are in the entorhinal cortex and place cells are in the hippocampus. And a place cell is actually just made up of a lot of grid cells, which are part of multiple modules. So there's multiple grid cells in each module and there's multiple modules in each place cell. And because each module represents like the periodic function of multiple grid cells, if you combine those periodic functions, it's no longer periodic. So that's why a place cell only fires at one specific location instead of repeating itself.
Yeah, and with this information, we can largely understand how the output patterns of grid cells, as pictured on the left, can lead to the output pattern of the place cell on the right. For future plans, so given this information and the programmed model we have of the head direction cells, here are some possible options for what we could do in the future. First off, we could create a model for the two-dimensional grid modules based on the ring attractor model that we have for 1D head direction cells. We could also implement a mechanism for combining outputs from multiple grid cell modules to generate a realistic place cell firing at specific spatial coordinates. So you could kind of model how it would look depending on where you are, and you could see the activity of place cells. And then along with that, we could test how environmental geometry transformations such as stretching or rotating space impact the stability and the structure of grid and place cell activation. Because as noted, everything shifts around when you shift from environment to environment, or if you even rotate. For example, head direction cells will rotate. So things can change the models. Here were some of our sources used, and thank you for listening.
Excellent, thank you. So this presentation was again around 11 minutes long. So great, very close to the time limit. Okay, so Geeta has a question. When people have vertigo, is there something that can be drawn from this information to correlate or cure?
I'm personally not sure the specifics of how vertigo occurs. I would say most likely, given what I know, this is related to vertigo in some way. Maybe the cells are damaged in some way, and so then you could fix some sort of system in the brain, but I wouldn't have a concrete answer on that.
Yeah, I don't know if vertigo is related to the cells used for navigation. Um, but if it is, then uh, maybe by repairing those cells, there could be like a cure or something like that.
And most likely, the cells involved would be head direction cells, at least from our presentation, even though there are many others that could be related.
Yeah, so just to add to that, people know about the vestibular system, okay, which is in the inner ear, is uh, which allows us to maintain orientation. Okay, there are some fluids in the inner ear which allows us to have a sense of where we are looking at and what angle we are looking at things in the environment. All that information actually has to go into the brain, and the brain has to process that information. And this processing of the information in the brain is what they are talking about. Vertigo, I think, has to do with the problems in the inner ear. I think. Um, uh, and they are talking in exactly how is the orientation and the spatial location encoded within deep within the brain. Okay, so yeah. By the way, this discovery, which won Nobel Prize in around 10 years back in 2014, um, was very interesting. Geeta says yes. They talk about crystals. So I was wondering. Yeah. So the connection is that from the crystals in the inner ear, you go to the brain, and that the brain part is what they were talking about. Um, yeah. What's interesting is where these grid cells and place cells are found. They are found in the hippocampal and area and the endocrine cortex, and why it's interesting is because these areas are usually associated with memory, and so the places where memories get stored and where local navigation happens is the same area. Okay, and so and finally it's beginning to make sense why um, if people have to memorize a lot of things. So there are people, for example, who memorize 100,000 digits of the digit pi, for example. How do they do that? One common technique is what's called memory palace. So they actually imagine that they're moving around in an environment, and they're seeing these different digits or different things, and that allows them to memorize long sequences of lists. And now we are able to understand exactly why it it makes it makes sense, because the navigation system and the memory systems are in the same area. And that's why when we say, if we think physically, if we actually imagine moving things around and we have a mental image of concepts, then it forms a stronger memory. And this is and again the reason is because it's all in the same area. Anyway, so that's just a side note. Any questions on the presentation? I should also add, like, there was a question asked earlier, how how is this B stuff related to how humans do things. The head direction cells, place cells, grid cells, initially found in animals, have eventually now been found in humans as well. Okay, so
Yes, I observed that same ring concept was also introduced in this slide. So yeah, there is some
exactly.
correlation. Yeah, thank you.
So it's the same thing across a lot of animals, from bees to mice to humans.